I can only say that I hope the Gadigal people had more luck getting to Darraglands than I did this afternoon down the M4. It was a shocker. And I just got to tell you one brief story. So I don't know which order to do this. I've got a talk, a rant and a story. And I think if I start with the rant, you won't listen to the talk because the rant's important, so I'll leave it to the end. And the story is that when I was a medical student, maybe an intern, I visited Great Ormond Street in London where Professor Elliott was a registrar in paediatrics. And we were at the Lamb and Flag having a beer at the same counter. And she started to tell me that she played a bit of squash. Now, I was a really good squash player when I was a medical student. I said, "You fancy a game?" And she said, "Well, all right, you look a bit young and you look like you're a man, but I'll still give it a go." She beat me 9-0, 9-0, 9-1. And that 1 point I got in the third game, I want to put it on my CV because I was prouder than almost anything else I've ever done. Anyway, my talk is about the passion I have for being a clinician researcher. And it's in cardiology, but you'll see that the lesson is equally applicable to everything. And I'm going to tell you a little bit about what I've done, but more importantly, what I've learned that kind of I wish I'd known when I was 20. So I was able to do some research along the way, which I'll describe briefly. Some of it in cells, which is on the left, a lot of it in humans, and some of it in the third world, or what I shouldn't call the third world, I should call the developing world. And each of them have been terrific, and sometimes we've been able to marry the two, and that's been really cool. So for want of a better term, the pearls I would like to say I've learnt over a long time is if you do a PhD, afterwards aim high, go for common diseases and go for high impact things. You know, once you've got your PhD, because a PhD is a bit different, you have to produce 3 or 4 papers. And so you can't really afford to deviate too much from a structured pathway of research. Because if you want to finish in 3 or 3.5 years, you've got to do stuff where whether the results are positive or negative, or positive or neutral, it'll be a publishable paper, and you've got to rely on your supervisor and your colleagues to tell you what you can deliver in a reliable way in 3.5 years. But when your PhD is over, you have a decision to make. Do you want to publish, 5 medium grade papers a year or 1 great paper a year? And of course you have to mix it up a bit. You still have to publish a bit to get grants, but you can afford after your PhD to aim high. And the benefit of aiming high, you'll be dragged down all the time because people will say you need to have 50 papers by the time you're 35 to get to be an associate professor. But 1 great paper will have impact beyond your place and time, and I'll show you which one it was for me when I was in my early 30s. And I'm really pleased that I went for something that was a bit riskier because it helped me with my career. And for me, it was the New England Journal of Medicine moment, and I'll show you the paper I did, and I got very impatient, and when they'd had it for 4 weeks, I rang up and said, "Will you publish my paper?" And they said, "Bugger off, we publish 1.3% of submissions, so call back in 6 months and we'll reject you." But when they finally said yes, it was really worth the risk. Second kind of pearl is it's really important to work with people you like. Now, how do you know whether you like them? Well, the best predictor of success is success. So begs the question, which some of you may have to answer in about 15 years, how do you ever get your first PhD student if you haven't got a track record of having successful PhD students? But in general, if you look at the evidence, the best predictor of success is success. If you have a supervisor who's supervised 10 medical graduates to PhDs in under 3.5 years, they're going to supervise you to your PhD in under 3.5 years. But it's also really important to have someone with empathy for what you're going through. And honestly, there are a lot of people out there without empathy. And empathy is not, like, I can't tell you the formula, but it's bloody easy. You know who's empathetic within a minute of meeting them because they ask about you rather than talking about themselves. It's possible to talk about yourself and be empathetic, but it's a better clue if they ask about you. So when you're trying to choose your supervisor, it's someone with a good track record of getting someone through their PhD in a timely manner, and it's someone who you think will be nice to you, because there are going to be times during a PhD, for those of you that go down your path, the low is usually at around 15 months, because you've spent about 12 months setting everything up, getting all the ethics and governance approvals, proving up your experimental models, and then there's a lag between when you do that, when you feel you're achieving stuff, and when you start to generate results that are meaningful. So at around the 15-month mark, people are usually a bit low and saying, what the hell am I doing that for? And that's when you need someone to take your hand and show you, that there is a way through to the promised land. Something that someone told me really early on, in fact, it was a really bad guy. I won't speak ill of people who've passed, but it was a bad guy, and he gave me great advice. And that was to maximize effort to reward ratio. In fact, what he said to me is, don't read a single word that you don't have to read, because we are in the information overload stage. So just look at every document you're given and say, do I really have to read that? And if the answer's no, just don't read it. So you have to reduce the time you're wasting. A lot of time is wasted reading beyond an abstract. Sometimes you do have to go into detail, but very often not. The second is there is a huge waste of time getting HREC and governance approvals. So utilise, I don't mean that HREC approvals are unimportant. Sorry, Human Research Ethics Committee. Pardon me, please pull me up on that sort of stuff. I'm empathetic, but I'm dumb. So this takes a great deal of time, but in most of the clinical schools, there is help available to you to navigate those processes. I say low success rate funding schemes. What is a high success rate funding scheme? You will know what the really low rates are. So I was approached today. Do any of you heard of a model called Helena Christensen? She used to go out with Michael Hutchins. Yeah, that table has. So she once famously said, I won't get up out of bed for less than $10,000 a day. I won't put pen to paper for a grant worth less than $100,000, because it's just, low success rate, low funding schemes, people will try and persuade you to do them. Don't. Okay, my personal one is focus on humans. Now, that's not to say that research on cells, molecules, and animal models are not very valuable, but at some point, a human is going to cross your path as a research subject, and that will be a joyful day. Okay, funding. Now, this took me a little bit of time to work out, but philanthropy can be a very good source of funding, and philanthropists care a lot about the disease that their children have. So sometimes you have to do a bit of due diligence. I think Professor Bauer has a fantastic funding stream, because obesity is A, extremely topical, and B, there are some extremely rich pharmaceutical companies that care about obesity. But if you look hard, there's a philanthropic group who will fund almost everything. So I've had philanthropic research from people interested in wet macular degeneration and people interested in the environment. And I've invented projects just because I knew there was a $1.5 million pot of money to do that project, which actually has, I've recently got a lot of philanthropic funding to study the environmental cost, the carbon cost of various health interventions. Now, that's not my main game, but it's become my main game because it's really interesting, it's really important, and there's philanthropic funding. And don't forget the disease-specific ones. There are a lot of funders that like a particular disease, like leukemia or diabetes, and that's almost invariably because of a personal connection. So just a few little examples of the fun that you can have in research, because it's not always intuitively obvious why research might be fun. And I've been involved in studying a disease called atherosclerosis that you all know very well. And on this slide, I just want to show you that a couple of the key events in atherosclerosis are monocytes sticking to the endothelial cell layer, which is there in red.
You need to advance.
Oh, I'm sorry. Okay. Got this confusing display with the before and after, and as you've already seen, I'm easily confused after an hour in Darug country. So monocytes stick to endothelial cells, and that's a pretty important early event in atherosclerosis. And then they get into the arterial wall, and they turn into macrophages which ingest oxidized low-density lipoproteins, and they become foam cells. And so this is an example of a study that Jane McCrohan did during her PhD with us, where she found, we wondered why men get more atherosclerosis than women, which is true even if you adjust for smoking levels and cholesterol levels and diabetes levels. And what Jane found was that these white cells, here the white cells are the macrophages and the black cells are endothelial cells. So lots of white cells sticking to the black cells is bad, and Jane found that if you treated these macrophages with testosterone and the endothelial cells, a lot more stuck. But every second week Jane would come in and say, the experiment didn't work last week. And then she'd come in and say, the experiment worked. And then she'd say, the experiment didn't work. And finally, we had a penny drop moment, and that was that when the endothelial cells were boy endothelial cells, treating them with testosterone made them sticky, but if they were girl endothelial cells, they didn't. So this is the effect of dihydrotestosterone, which is a powerful androgen, on monocyte adhesion to endothelial cells. And this is why Jane's experiment only worked half the time, because when the endothelial cells came from women, there was no effect. When the endothelial cells came from men, there weren't. And she went on and found almost exactly the same with macrophages, but by then the penny had dropped that you use male macrophages. And what Jane was able to find in her PhD was that if you treated macrophages with androgens, with testosterone, they gobbled up a whole lot more cholesterol and they turned into foam cells, which is a bad thing to have. And then she went on to look at what androgens do to the genes inside of macrophages. And I tell you, I'd almost think that she'd made this up, but I don't think she had. This was part of Martin Ng's PhD. When she looked at the genes upregulated by testosterone in male versus female macrophages, there were all of these that were upregulated in the male macrophages and all of these that were upregulated in the female macrophages. Now, you almost never see sexual dimorphism like this. You almost never see a result like this, which is why I think that Martin might have made it up, because he's a suspicious character. But it's been replicated by other labs, so he probably didn't make it up. But it was a very cool piece of research. And so sometimes mixing common sense, serendipity, and basic science gives you really important insights. Now, the next thing that's kind of obvious, but it's worth stating, is that common conditions result in high-impact publications. So this is a piece of work from my PhD where we showed that smoking, so FMD is a measure of arterial health. The higher your FMD, or flow-mediated dilatation in your artery, the better your endothelium is releasing nitric oxide and the healthier your artery is. So higher number, better. So this is endothelial functioning controls in current smokers and in former smokers, and that got into a high-impact cardiology journal called Circulation. But then we went on to look at it in passive smoking. So common disease, topical thing. And this is kind of really cool. This is a bit the holy grail of research, I think. And I'm not being boastful, I'm just saying we were right place, right time. So what this study was looking at endothelial health in teenagers who were passive smokers. So these were teenagers that didn't smoke themselves, but they were in a family where both their parents smoked. And what you can see here, and I'll tell you why I'm presenting the detail in a moment, is that if you look at the passive smokers, their arterial health was bad. They were 15-year-olds. It was as bad as the active smokers, which is a bit counterintuitive, except they had been passive smokers for all their life and the controls had only been active smokers for a couple of years. And there's something very interesting and noxious about passive smoke or environmental tobacco smoke, and that is, as you probably know, when a smoker inhales, the tip of the cigarette goes red, and that's because it's combusted at very high temperature, whereas passive smoke isn't. So there are a lot more toxins in passive smoke, and it's not filtered. So passive smoke is really bad for you. Now, the interesting part about this is a study on 78 teenagers in Sydney, Australia. Some guy in California read this in the New England Journal of Medicine, and he happened to be a congressman, and he was a congressman on a mission. And he took this and another piece of research about the effects of passive smoke on asthma in children, and he went to the California legislature, and he said, hey dudes, we got to stop smoking in schools. So they did that. And then they got excited and said, we're going to stop smoking in pubs and clubs and in hospitality venues and hotels. So California became the first jurisdiction in the world to ban passive smoke or environmental tobacco smoke in those places. And then, to cut a long story short, it was taken up almost all over the world. And this little piece of research on 78 Australian teenagers has been estimated to have already saved 45 million lives. 45 million from a study on 78 kids that you could do in your backyard. So, you know, it's kind of important to study a common thing, but if you get it right, you can have impact genuinely. You know, this research has influenced people who I've never met in countries that I'll never be to. And so you can have outsized impact. And just because I knew that Professor Bauer was in the audience, we've done quite a lot of work on childhood obesity and the effects on the vasculature. And this is kind of similar. You look at a common problem at a stage that's a little bit counterintuitive. You know, who knows that 11-year-olds get atherosclerosis? But they do. And we did a couple of studies I'll show you very briefly, where we found that mild to moderate obesity in children leads to arterial wall thickening and impaired endothelial function, which is a bad thing to have. And again, just 36 kids. And we found that the arterial walls were much thicker in these children. And we found that the arterial function was much worse. And so we went on with a colleague in Hong Kong, where we ended up doing this study, looking at 82 obese children and randomising them to diet only or diet and exercise to see what that did to their blood vessel health. And slightly complicated slide, but remember, the higher the number, the better. So with diet only at 6 weeks, the children got a little bit better, but with diet and exercise, they got a lot better. But if you wanted the benefits sustained at a year, you had to do long-term exercise. So there was no shortcut, and I guess that's not a big surprise. But again, to make the point that relatively modest research goals, if you get the question right and design the experiment correctly, can give you massive public health benefit. And then we went on to look at passive smoking and reversibility. This was the good news, that if you could take a teenager away from the passive smoking environment, their arteries could recover within 2 years. The last piece of research I wanted to share with you, or type of research, is developing world research. And have any of you heard of a wonderful professor here, Clara Chow, who's Professor of Cardiology at Westmead? So Clara is just all kinds of wonderful. And when she was doing her PhD with us, she got on a plane to Hyderabad, and then she got on a train to Godavari, and then she got on a bus to West Godavari, and then she got on an ox cart, and I went with her for the first trip, and then she was on autopilot. And at 6 a.m. in the morning, before they all started working, Clara went and studied these people for effects of diabetes and atherosclerosis. And what she was able to find was high levels of subclinical atherosclerosis and a 13% rate of type 2 diabetes in adults in West Godavari village. So this was like the place that God forgot or remembered, depending on your perspective. This was a place where we thought Western-style risk factors didn't exist, and yet diabetes and atherosclerosis were already extremely prevalent. And I've had the good fortune, and it's deliberate, to do studies in China. I thought, talking about innovative ways of getting funding, we called this study the Cathay study, Chinese atherosclerosis in aged and youth. And we thought Cathay Pacific would sponsor us. Did they? No, but the name still stuck. But it was worth a college try. We did Hindi studies in India, beautiful studies with colleagues in Mozambique and Cambodia, which I'll discuss in a moment, and a really cool malaria project, although this was a bit of a trap for young players. In Indonesia, there's a lot of malaria. Believe it or not, malaria is a vascular disease because the parasites sludge in capillaries in the brain, and that's what causes cerebral malaria to cause death. So it's actually a problem of ischemia related to the parasitized red cells. And this project was going absolutely fantastic with Sinyo and Nick Anstey and me. And then the government changed in Indonesia, and they decided that Australia were kind of what Donald Trump thinks of us, just not worth bothering with. And ethics permission was withdrawn from the study simply because we were Australian. And that paused for 3 years, but then it got going again. So developing world research, not necessarily that easy, but worth doing. And here's an example of the data, the kind of data we were able to generate. And again, you'll see the lesson from it, I hope, in a moment. So this is a study called the Prevalence of Rheumatic Heart Disease by Echo Screening. And we compared an expert with a stethoscope to someone who'd trained in echo for 4 weeks and went out into schools in rural Mozambique and rural Cambodia. So 2 studies of over 10,000 school children, and we compared the prevalences of early detection of rheumatic heart disease with clinical versus echo. So what does this mean? Well, if you detect an 8-year-old with rheumatic heart disease, you give the kid penicillin and they won't die from rheumatic heart disease when they're 20. So early detection is really important. And what this showed was that you could take someone, train them for 4 weeks, give them a portable ultrasound machine that costs $10,000, send them to schools, and they would save a lot of lives. So we did this research, and then I found a wealthy philanthropist in Sydney who cares about Vietnam, and he gave us $50,000 to set up a program for RHD screening in Vietnam. So again, if you're thoughtful and you're strategic, you can make massive impact on important health problems across the world. Okay, I'm on the home stretch before my rant. So I am a cardiologist. I do adult congenital heart disease. I have lots of patients, many of whom are very sick. And I think in clinical, there are 2 ways to help be a clinician-scientist to allow you to do time to do your science, because you need a clear mind and you need a headspace. So one is to train colleagues to share the clinical load. So I've had the great good fortune to go on a few sabbaticals for 4 months, and I have 2 terrific people who do what I do in cardiology and who help out when I'm away, and I help them out when they're away. The second is to make sure your head of department believes in shared service rosters. So you're on for a week, and then you're off for 3 weeks. You don't look after your own patients all 4 weeks. You have time free of clinical work so that you can do your research. We've touched a little bit on the maximising effort to reward ratio, reading less, try and get a relatively low-cost admin helper for your ethics committee work and grants. And the other thing that's really cool is whatever else you're doing, be involved in teaching, because you become a much better thinker if you're a teacher, particularly with what I've called the Socratic method, which is where you take a case and you debate it. It's not didactic, it's interactive, and that makes you a better teacher and a better researcher. So I've been very lucky, as will have been evident, to be able to do these basic clinical and developing world things. But it's all been very deliberate and strategic. I sort of sat down when I was 28 and thought, how do I want to conduct my research life? And it was like, when you have kids, you don't read the paper in the morning. You take them for baby chinos and you read the paper at night. You can apply similar strategic thinking to work-life balance when you have your own family, if you have your own family. So it's possible and it's cool. Now, I'm going to take, and I've got 1 more minute for a rant. Okay, my rant is this. My rant is that everyone is prolonging your training time. And that makes it harder to be a clinician-scientist. So you've got to start earlier. So I started, and I'm sure Elizabeth and Louise started medical school when they were 17. I finished when I was 21. I'd done my PhD by 28, and I was a consultant when I was 30. That's not because I'm good, it's because the system allowed it. Now, the average age of starting at a graduate medical program is 24. The pass rates are a little lower. Basic physician training, for example, is 3 years rather than 2. Advanced training is 3 years rather than 2. So you end up being a consultant when you're 97. The Australian community don't get much value out of you when you're 97. But more importantly, the age when you're starting a PhD is also an age when you might have a young family or want to have a young family and have a mortgage. So I am on a relentless campaign with the University of Sydney and the College of Physicians to shorten everything back. Because I think if you gave me a smart kid at 18, I could turn them into a very good cardiologist by 23. And I really don't know why it takes 17 years to do that, but at the moment it takes 17 years. So we've all got to lobby people to try and shorten what can be shortened. I don't hear people saying Australian doctors are badly trained because they did 2 years of basic training and 2 years of advanced training. I have no idea, other than money for the College of Physicians, why it's all been prolonged. But that's why I ended with the rant, because I didn't want to depress you. What can you do about it now? What you can do is plan early. Do your PhD while you're doing your advanced training. Do a 4 or 5 year program of doing both at the same time, which requires a good supervisor. But just be aware that time is a little more against you than it was for us. And again, you have to think strategically how you're going to overcome the time penalty, because being a clinician researcher is simply the best. You use your clinical knowledge to inform your research questions. You use your research finding to inform clinical guidelines, and it's this most wondrous, virtuous cycle. And I really hope that any of you who wish to do it find the opportunity to do it, but it will require a little bit of strategic thinking. Thank you.
Thanks very much, David. Have we got time for a couple of questions, Catherine? Yes? Anyone got any questions for David? Well, we've got him so far out of the eastern suburbs. Louise has always got a question.
Thank you so much, David. I was wondering if you could just talk about how you might, for example, do a PhD and do advanced training, and issues to do with getting scholarships and things like that. What was happening then, what you understand now?
Yes, so for example, in our department, we have a 5-year scholarship program which gives 2 years of research funding and 3 years for advanced training. But there are mechanisms for funding through, and they're sort of well advertised through universities, through colleges, through philanthropy, etc. So again, it's hard for people before to understand those, but supervisors should understand those. So there are funding mechanisms, both project-specific and person-specific.
Thanks, Louise. Any other questions for David?
Thank you for your talk. My name is Annie, and I'm from the University of Sydney. I just have a question for your project before, like in the rural area where there's 13% of the population still getting like the diseases from the Western world. What causes that 13%?
So there's a simple answer and a complex answer. The simple answer is it turns out that South Asian Indians are more prone to metabolic consequences of obesity at a lower body mass index. So for example, the healthy body mass index in Caucasians is 25 or under. In South Asian Indians, it's 23 or under, because there's a genetic predisposition to visceral obesity rather than subcutaneous obesity. That's the simple answer. The complex answer, which is why are they more prone to visceral than subcutaneous obesity? I don't know. There is someone here who probably does know, but I don't know.
Okay, here first and then...
Annie, the icebreaker, has done a great job.
I'm Ellen from UNSW, and I was just wondering, like, getting to that position of being a clinical scientist, how does that differ from being a medicine student or a medical science student? Okay,
so in fact, being a medical science student gives one a great advantage for a career as a clinician scientist. So I probably should have defined the terms. So a clinician scientist is someone who's a specialist or a GP, like who's finished their training, but who devotes a reasonable proportion of their time to research. So, for example, university academics, for what it's worth, are encouraged to do 40% teaching, 40% research, 20% leadership in their research time. So it's mixing. So most doctors that you meet are purely clinicians, but there is a subset of clinician scientists who spend part of their time doing clinical work, part of their time doing science, and they're clinician scientists. And I think to your question, you have to kind of be trained in both. So in a way, doing a medical science degree before a medical degree gives you a great springboard to become a clinician scientist because you have a better understanding of clinical science.
Actually, not a question. It's just a follow-up on your work on passive smoking. Just wanted to remind you that you didn't only collapse the business in California, you did in Ghana as well, which led to a total ban on public smoking in the early 2000s to mid, but also cost my uncle his job because he was the country director for British Tobacco when it collapsed.
So I'm sorry your uncle chose that job, and I hope your conscience is not too guilty.
No, not at all.
Well, look, I think we can all see why David was a world debating champion. He's a great communicator. I just wish I'd heard some of your pearls earlier, David. I think I've read far too many words and applied for far too many low volume, low whatever it is, grants. But what I didn't give you was David's statistics. No, he's 5'10" and 80 kilos. No, he's got an H index of 110. That means that he's had at least 110 papers with 110 citations in the literature, well over 100,000 citations. And but I think what's much more than that, and hundreds of papers, is the impact that he's demonstrated, and really that's what we're all aiming for. And particularly as a clinician-scientist, you have the opportunity to see the impact in patients and populations, and also to ask the questions when you're in clinical practice. What are the important questions begging that might warrant some research? So I think it's been a fantastic talk, David. Thank you so much.